Welcome back, and today we're going to work on bacterial genetics and gene control in prokaryotes. So the first thing we're going to look at is a bacteria cell, and the genetics of it's very different than the eukaryotes that we've been looking at so far. And the main thing is, instead of having, in the case of humans, 46 chromosomes, they only have one chromosome. So they have this single loop of DNA located in the nucleoid region. And many of them also have small bits of DNA, little loops known as plasmids. And these are going to be very important as we do a lab later in the unit, but also as we go through this and look at the different genes and traits that can be passed on separately from the bacterial chromosome. Plasmids exist and replicate independently of the chromosome themselves. So a bacteria cell may have its own DNA, its own chromosome, but also several little bits called plasmids. One example that we'll look at in greater detail a little bit later is the fertility or F factor. So this plasmid here, again, it's a loop structure, and what it does is it encourages cellular attachment during this process of conjugation, where bacteria cells can actually exchange some DNA. It's a one-way exchange, and in this case we're going to be looking at what's the F plus plasmid or the F plus gene, and that's the donor that can actually transform other cells to become from F minus to become F plus. So here's an example of a plasmid that we'll look at in a little bit more detail. So again, bacterial plasmids, loops of DNA found in some bacteria. They contain special genes that are not related to basic life functions. They don't need them, but they can certainly help. One example is antibiotic resistance, so ampicillin, MRSA you may have heard of. They can be transferred from one bacterial cell to another. So here's the bacterial DNA in red that has all the life controlling genes, but they may have one or several plasmid DNAs that have different genes on them as well. So, replication in bacterial cells is very similar to what we saw in eukaryotes. They use a lot of the same enzymes, and the process in terms of the replication fork is the same. But because they only have one loop, that plasmid, there's only one origin of replication, and from that point of origin replication, the replication fork continues just in two directions. So rather than having many bubbles like we saw in the eukaryotes, the prokaryotic ones just continues all the way around, and it reaches the termination. And bang, we've got two new loops of DNA. The cell will then split through binary fusion. It doesn't use mitosis or meiosis, and we've got two new bacterial cells. So this is one of the reasons why bacteria can reproduce so quickly. In some cases, as quickly as every 20 minutes, they can double in numbers. One way of looking, or one way that the genes can be altered, so the major way that bacteria build up genetic changes through mutations, but they can transfer DNA, so they don't have the benefit of carrying out meiosis and getting genetic recombination like animals and plants, but because of transformation, transduction, and conjugation, they can alter their genotypes. So the first we're going to look at is transformation. And this is the alteration of the genotype by the uptake of naked foreign DNA. It's live, non-pathogenic cells will take up foreign DNA and incorporate them into their chromosome or plasmid. So they're taking DNA from another source, incorporating it into plasmid, and then expressing those genes in the future. So here's a quick and easy picture of how it can happen. So here's one bacteria that has been lysed, it's broken up, and some of its DNA has entered through the plasmid membrane and become incorporated into the new genotype, into the new plasmid of that bacteria. So here's an example of bacteria where we've got two mutant strains. They need both arginine and tryptophan in order to survive. And so here's one mutant strain on the left that can make arginine but not tryptophan. And so when we grow that on this minimal medium, which basically just means it's got some sugars and salts in there, it doesn't grow. There's no colonies. It can't make those two amino acids that it's need. And over on the right, it's kind of the flip. It can make tryptophan but arginine, and we get the same case. We culture them, we grow them on the minimal medium, and we get no colonies growing. But when we mix the two strains together, there is a transformation, so there's an exchange between those two strains, and we get, as you can see here, the purple colonies that can produce both arginine and tryptophan. So a transformation, an exchange of genetic information or material has taken place between those. The other case that we've already seen is with Fred Griffith's experiment with the mice, where he took the live virulent strain of Streptococcus and injected into the mice. The mice died. He took a non-virulent R strain with no capsule around it, injected it into the mouse, and the mouse was fine. He then took the virulent S strain, heated it up, killed it, injected it into the mice. No effect. The mice were fine. But when he mixed the two, both the ones that led to healthy mice, the live R strain and the dead S strain, he injected into the mice, and the mice died. So he concluded that there was a chemical substance from one cell is capable of genetically transforming another cell. So the virulent DNA from the dead cells was somehow transferred into the non-virulent R cells. The next one we're going to look at is transduction, and here is where viral phages carry bacterial genes from one host cell to another, so it actually needs the virus as the vector to transfer the DNA from one cell to the other. And there's two types. There's generalized, and in this case the bacterial DNA is packaged into a viral capsid, so it kind of gets packaged wrong. So you get the entire bacterial DNA, and it goes off in a virus, and then there's specialized, and this transfers only a restricted set of genes along with viral DNA, so it's kind of a mixture. So if you look at the diagram here, on the left here's generalized. So here's our bacteriophage infecting the bacteria. And when the host DNA is hydrolyzed, it's chopped up into pieces, and we start replicating the viral DNA and proteins. When they're repackaged, a bacterial DNA fragment is packaged in a phage capsid. So here we get basically a virus with bacterial DNA. When that phage goes and infects another bacterial cell, it can inject that bacterial DNA, and in this case we have a cell that is being transformed from its original A- genotype to an A+ genotype from the gene that it got through the phage. Specialized is similar, and here is when the viral prophage is incorporated into the bacterial cell, and we may have, again, here's the A+ gene, and now when the new phages are made, occasionally a prophage DNA exits incorrectly, taking a joining bacterial DNA with it. So instead of being entirely bacterial DNA like we saw on the left, what happens is we have mostly viral DNA, but we have a small piece, in this case the A+ gene of the bacteria, so that's why it's specialized. And when this phage infects a cell, the prophage gets worked into the bacterial genome, and the A- gene can be replaced by the A+. So in both cases we have recombinant bacteria. We have a new combination of genes that wasn't seen previously. The last step we're going to look at is a process called conjugation, and this is the direct transfer of genetic material between two bacterial cells that are temporarily joined. So here the DNA transfer is one way, and they attach by this structure called a sex pili, and so there really aren't male and female. We'll call one of them male, and as we'll see in a minute, those are the ones that will have the F+ gene. And so the F factor for fertility, as we mentioned in the first or second slide, is mostly what we call the male. It's going to be the donor, and it will transform F- cells to F+, and it's on a separate plasmid. And then what we'll see is if the F+ integrates into the bacterial chromosome, the cell becomes HFR, which is a high frequency of recombination, which allows it to transfer its genes to other bacteria more readily. And the last one that we'll look at here is just real quick the process that happens. And we can see on the upper left here, here's the F+ cell. So this has the F plasmid, and cells that don't have it are F-. And so here is the mating bridge of the sex pili, and what will happen is there will be a transfer. We'll copy that gene, and that new plasmid now is in the previously F- cell, and we now have transformed it into an F+ cell. Now, it may stay that way, but if the F+ gene becomes incorporated into the host's chromosome, it becomes what's known as an HFR, or high frequency recombination cell. HFR cells can then go and conjugate with F- cells, and they will transfer that section of the DNA along with others, and will turn it into an HFR cell as well. and we can see the recombination between the HFR and the F minus, and so that we get a recombinant, and in the case right here it's the B plus gene that has come, and the original F minus cell was all C minus, D minus, A minus, B minus, but the B plus that came from the HFR cell has now recombined it. So just briefly, this is how bacterial DNA replication and transformation, transduction, conjugation works. And now you're going to look at a couple animations and answer a few questions about them. Thank you.